So passing on to you.
Thank you very much, Jeff, for the invitation, and it's my pleasure to talk to all of you. Here are just some questions to start off, thinking about as we go through the talk. How should we quantify uncertainty for black box ML algorithms? When we use ML algorithms in practice, how do we go beyond point predictions and going to estimate uncertainty for these? It's called predictive uncertainty. And so, I also work on distribution-free uncertainty quantification via something called conformal prediction and post hoc calibration for classification. These will actually be able to fit into this talk's framework. So even though this talk will be about experimentation more from the testing front, you could also be doing experimentation from a prediction front, and then slightly different tools on which I have papers as well would be able to fit into the general broad framework that I'll be working in. This is what the talk will be mostly about today, is how do we quantify estimation uncertainty in sequential settings. We often stop our experiments adaptively at data-dependent stopping times, and we want tight confidence intervals, for example, for the lift that you get or an effect size at those stopping times, and the usual confidence intervals we construct are not valid. And so, we typically solve these with certain martingale approaches to quantifying uncertainty. And these can also be thought of game theoretically as playing a game like the statistician is playing a game against nature. And you can view both testing as and estimation as betting against nature. Again, a lot of this talk will be motivated by this type of a question. How do we avoid selection bias and false discoveries when we test thousands of hypotheses? So this happens both in science and in technology. In science, often, every gene is associated with a hypothesis, like is this gene associated with diabetes, but we have tens of thousands of genes. And we are often, testing all of these because we don't know which of them are related or not related to diabetes. In tech settings and IT settings, things often proceed sequentially, where all the hypotheses are not written down at one go, but every week, or maybe a different person in the company might come up with a different hypothesis of something that might improve the lift, and they might want to test that hypothesis, obviously, before actually making the change. So the multiple hypothesis testing that occurs is much more subtle because it's not occurring all at one go. It's occurring, over time. And so we will talk about that. There's several error metrics that are popular. The false discovery rate, that's what this FDR is, and various types of structured settings where you might want to take into account the structure if you know it, or prior knowledge if you know it as well. And in some sense, this is all in maybe a slightly broader theme of how do we enable interaction between the scientist and the algorithm. We don't want to entirely, exclude ourselves from the process, maybe because we know notions of structure or prior knowledge or things of that kind, and we know at a higher level what things we're doing. The algorithms themselves maybe are more narrow and can give you a particular level of inferential statements, and we want somehow us to be involved in the decision-making process. Is somewhat interactive, but if you just interact in an arbitrary way, then it causes selection bias because you're looking at the data that's and making decisions based on that data, and so it causes selection bias. And so the question is, how do you protect yourself against selection bias and so on? So these are just some high-level motivating themes that I'd like to explore in this talk. Here are some slightly more specific questions. So I might have written them in a language that may be more familiar to the sciences, but this is simply because I want you to be able to think of not only, I'm sure you'll be able to connect it to experimentation in the tech industry, but I want you to be able to imagine these questions in fields outside of tech, like in the sciences, because there are a lot of commonalities, so that when you see perhaps papers written with a scientific perspective on it, you'll be able to realize, oh, actually, it makes the same question make sense for tech as well. So here's the first question. So in any scientific subfield, do the relevance and importance of the claimed or published discoveries that people have made, do they depend on the order in which hypotheses are tested by various scientists over time? And should the importance or relevance of those discoveries depend on the order in which they're tested? We like to think about science as the pursuit of knowledge. But if actually the relevance and importance of some discoveries depend on the order in which they were discovered, but if they were discovered in a slightly different order, you wouldn't think of them as being as relevant or important. It's a bit odd. Because we like to think today of science as a set of things we have found over the past, but maybe it really depends on the order in which we found them. Now, the same question obviously applies in tech as well. You have a particular maybe setup of the website or app or whatever it might be at the current state, and you have tried to optimize certain metrics over time in different parts of the website, but maybe the current state of the website is very dependent on the sequence of things that you did over time, and maybe if things were tested in a slightly different order, the same things were tested in a slightly different order, you might find yourself in a completely different state of affairs at the current point. And so if that's the case, then it's at least something to think about, like how much has this order affected things. A second question is, for large publicly shared scientific data sets, this could be in genetics, neuroscience, the heart, things like that, is the statistical validity of proclaimed discoveries, is it affected by how many people have downloaded the data set and tested hypotheses on it? And is it possible for a data set to get stale? What happens when some downloads lead nowhere, but others lead to some interesting findings and are published? So this is a question more about post hoc analysis of data. So if a company has collected a bunch of data for a year, and then over time people be like, okay, let's do something with that data we collected. Maybe we ran some large survey, we ran a large test of some kind, and then we want to use that in some way to improve something in the future. So maybe the data set is made available to all employees at any time. Lots of employees download it. Some employees don't do anything after they download it. Other employees find interesting things about it and say, hey, I found something interesting on this data set. Let's act on it. So is it possible for data sets that are collected like this to get stale? Which means that as more and more people download it and try things on it, obviously they're going to find things. At what point should we say, hey, actually, we have like really squeezed out all the juice out of that data set. We should really not be trusting the discoveries made from it anymore. Here are two other questions. If we often correct for multiple hypothesis testing, so for those who are familiar with it, it's the notion of multiple testing is you're just testing thousands of hypotheses. So obviously many of them will appear to be true just by chance. So in science, you often correct for multiple testing, but that correction is done at a particular scale. Like for example, within one single paper, all of the hypotheses I test there, they are corrected for there within a paper. Or sometimes even within one simulation within a paper, I say, okay, wow, I've tested 5,000 hypotheses, I should correct for that effect. But why do we restrict ourselves to that granularity? What about other granularities? For example, if a lab writes multiple papers, they only correct within each paper, but not across papers. If there's a field with multiple labs, then they don't correct across labs. They only correct within each paper within a lab. And what about at a journal? So like at some level, the multiple testing issue, as you go up the hierarchy, a journal is receiving publications from many subfields, which each has many labs, which each has many papers being written, and the multiple testing correction is only corrected for within the papers. You zoom in the most and you correct over there, but it's really not corrected at the larger scale. So the question is, if you should correct for it, then how should you correct for it? And if you say, no, we don't need to correct for it, we only need to correct it at the unit of a simulation within a paper, then why? What's the justification for why you can ignore the larger scale multiple testing effects? And maybe this is the most direct way the last question is the most direct way to think about the issues that might arise. So let's say that you want to test 100 hypotheses. So instead of testing all 100 of them today, if you've tested all of them today, you'll be forced to correct for multiple testing, because there's now a chance that out of these 100, some of them can be true. You can reject the null hypothesis and proclaim a discovery just by chance. So you would have to be forced to correct for multiple testing if you were testing all 100 today. But then maybe instead of that, you want to avoid that. So what you do is you randomly order the hypotheses and you test one today. And you say, oh, I'm only testing one, so I don't have to correct for multiple testing. Then you test one tomorrow. and you're only testing one, so you don't have to correct for multiple testing, and the one the day after, and so on. And you do this for 100 days. And so since each day you only tested one hypothesis, you don't have to do any correction, and so you've gotten away with it. You've tested the same 100 hypotheses, but you just did them one at a time. So now, like, if you zoom in to that lower level granularity, there was no multiple testing correction needed, and you've escaped this. So this seems paradoxical, and it is. And my personal argument is that even when the hypotheses are tested over time, you still need to acknowledge and correct for the multiple testing effect. Otherwise, you're just going to, at the end of the year, and you look back at the experiments that you have run a few thousand experiments, you're going to find many of them as discoveries just by chance. And if you don't correct for that, then you're going to be in trouble. So anyway, these are just supposed to be some thought-provoking questions. We're not going to particularly address any one particular question out of these. They should just make you think about various things about how discoveries proceed, what their implications are, false discoveries, and so on. So I think of science and often tech as being a sequence of sequential experiments. So there's not one sequential experiment going on, there's a sequence. So this is just a cartoon figure of what I mean by that. Like the x-axis is time, or the number of samples that you've collected so far. The y-axis is just the experiment number. And so there are no temporal or longitudinal effects that I'm going to talk about in this talk. Those can be talked about separately. Here I'm trying to make a different set of points. So the way it goes is, in the first experiment starts, and then at some point you realize, hey, actually there's something else I want to do, and the second experiment starts. And and time goes on like this. At any given point in time, for example, a third experiment might start, maybe it's not very promising, and you choose to end it early. And so the third experiment could have ended even before the first one ended. So there could be multiple experiments going on at the same time, just because different teams within the same organization have started these experiments to test different hypotheses, or even within the same team, different employees might want to test different hypotheses, so they might start their own test for that reason. And if something is more or less promising, you might want to run it longer or shorter. So maybe this experiment is really long because they're expecting a small but sustained effect, and you just need more samples to eke out that effect, and so on. So I don't know what the reasons are that these experiments were. Maybe 3 are started in parallel and ended in parallel. Maybe there's a mini batch of experiments running. The point is, it's some process like this. There's just some set of experiments running for different periods of time in parallel, and it keeps going on forever. Okay, and so this is a cartoon. These could be A/B tests. These are often used in the tech industry. I'm going to assume that many people here know what A/B tests are. These are sequential two-sample tests. Usually, A is the placebo or the current version you have. B is the new version or the treatment, and you want to test if basically is A the same as B, or is B better than A, and so you collect data sequentially until you can make this judgment. But the tests could be more complex than that. They could involve multi-arm bandits. They could involve some kind of policies. So the tests themselves could be more complicated. Again, it's just a cartoon to show you this. Something like this must be going on. It does go on in most tech companies. On the other hand, like in a pharmaceutical company, there could be this red line, which is a common control population. So you could imagine that, for example, take your favorite Pfizer or I don't know, something of that kind, and it's actually experimenting, let's say drugs for some disease like Alzheimer's. And over a very long period of time, it comes up with different, like researchers come up with different ideas, different treatments, different drugs, basically. And then these might be different sequential clinical trials. There's a common control population for, maybe these are either people without Alzheimer's who are given the drug to see some side effects, or there could be people with Alzheimer's who are given a placebo. But basically, there's like a common control population across all of these. And the treatments are the new ones as the researchers come up with it. Maybe we're not going to solve it anytime soon. So this process is going to keep going on for years, maybe decades, and so on. So like you can imagine this kind of a situation in a pharmaceutical company. But the unit of time is anything. So you could even have it in a tech company where you're not sure what the right thing to do is, and so you have a common control population, and you keep changing the treatment over time to figure out what's really going on. Okay, so the main question you would have is, in this kind of a setting of doubly sequential experimentation, what kind of guarantees would we like? Okay, so this is for the, I like to think about things first for the inner sequential process, and the inner sequential process is just one experiment. So if I zoom into just one experiment, what would I like from that experiment? I would like correct inference when the experiment ends. And so this could be, if you're testing a hypothesis, you want maybe correct p-values for that A/B test. If you want a confidence interval for the treatment effect, you want something that's valid at stopping times. So the inner sequential process, that's what you would like. For the outer sequential process, when there's multiple experiments increasing in number over time, and obviously when you're running your experiments, you have no idea how many future experiments are going to be done. So it's like less clear what kind of guarantee you would like for the entire overall sequential process. And because this is less clear, and because today we are myopic and we say, I'm running these 3 experiments right now, I have no idea what's going to happen in the future. I'm going to focus on these experiments that I'm running right now. Because we're often myopic, we tend to focus on the inner sequential process, and we tend to forget, you just ignore the outer sequential process. And that's not a good idea typically. So what are some potential issues within each experiment? So this will, of course, depend on the tech company or the pharmaceutical company and so on. But some, let's say, typical more common issues within each experiment are some kind of continuous monitoring might take place as the data is coming in. The data scientist is looking at the data to see how it's going, and might want to adaptively make some decisions based on the data that they're continuously monitoring. The second one is that the experiment horizon may not have been fixed in advance. So often with clinical trials, it is fixed in advance. There's an FDA oversight that you have to, when a clinical trial is being run on humans or anything like that, you have to tell in advance, okay, the first phase is going to have these many people, the second phase, these many people, something of this kind. And so there's some oversight. In the tech industry, there's almost no oversight. So in different companies, different employees run experiments for different horizons, and the horizon may or may not be fixed in advance and could be adaptively decided on the fly. So there could be arbitrary stopping or continuation rules for these experiments, and so that increases the amount of selection bias, because you're making data dependent decisions to stop early if you see some interesting evidence, or run the experiment longer if you haven't seen enough evidence yet. So these kind of data dependent decisions could cause issues if you don't realize that actually a lot of the confidence intervals or p-values that we teach are for fixed sample sizes. If you fix n in advance and you form your t-test confidence interval or a p-value, then it's correct. But that's only if you tell me in advance, I'm going to collect exactly 10,820 samples and I will not less and not more, then it's valid. But as soon as you say, well, I don't know how many points I'm going to collect and I'll decide as I see the data, none of those are actually correct. And so we're going to want to develop new techniques that are correct at arbitrary stopping times. Some issues that occur across experiments include selection bias in terms of what you want to report. In the sense, you run many experiments, but you just don't mention many of the things that you run. You only mention the ones that appear promising, and you don't mention the others. And so this selection bias of which ones you choose to explore further or act on and so on, that's what you typically correct with multiple testing. There could be dependence across experiments. If you're running experiments in parallel, then there could be shared people making actions and which affect data from multiple experiments. There could be some kind of dependence between the p-values or the confidence intervals. And of course, one of the main ones is we don't know the future outcomes. We don't even know how many experiments will be done in the future. So, how do we even correct for that? Many other concerns. I'm just giving you some examples of concerns within and across experiments. Okay, so I'll just, given this is a short talk, I've over time given 4-hour tutorials at KDD and stuff about this. So given the length of this talk, I'll just give you a brief glimpse into one solution for the inner process and one solution for the outer process. Okay, so and there's plenty of subtleties to talk about at length afterwards. So for the inner process, I'll introduce this concept called confidence sequence. And for the outer sequential process, something called the false discovery rate and so on. So let me just dive into it. Okay, so this is based on a sequence of works. If you're looking for through my publication list, there's actually a set of works outlined which is on sequential non-parametric testing and estimation. And so some of these are published at statistics venues, some of them at ML venues and so on. So yeah, and here are some of my excellent collaborators. I've particularly named the students, Steve Howard and Ian Wadby Smith, who have worked with me on these topics over time. Okay, so what is a confidence sequence? So most of you are familiar with a confidence interval. A confidence sequence is just a sequence of confidence intervals, ln un, where n is supposed to represent the sample size. So as I increase the amount of data, then I'm going to construct a sequence of confidence intervals, and they're going to have a simultaneous coverage guarantee. So it's not that just for one n, the probability that theta is in ln un is at least 1 minus alpha. I'm going to ask that for every n, theta lies in ln un. Okay, so this is a much stronger guarantee. It's really like a confidence band, if you want, over time. That's what I'm forming, not just a single interval. And so this notion was introduced, 50 years ago, but kind of lost popularity over time because in clinical trials, when it's so heavily monitored and you know exactly when you're going to stop in the 3 phases, you can do something slightly better. But in settings in which you want the flexibility of adaptive continuation and stopping of experiments, this is perfect. I'll explain why this is useful, but essentially it's because it's a continuous confidence band and not just a single interval that you're forming for whatever parameter theta that is of interest to you. Theta could be, for example, a treatment effect difference between a treatment arm and a control arm. So that could be theta. So remember the first part of the talk is for a single experiment. So I have one experiment and I want one treatment effect between a treatment and control, and I'm collecting data sequentially for that experiment. I'm just ignoring the other experiments for now. We'll come back to that in about 10-15 minutes. So for one experiment, that's a confidence sequence for a single parameter theta. So this is much stronger than the pointwise or a fixed sample guarantee that you typically find written in your statistics textbooks or blog posts or whatever. And the main difference is the for all n is actually outside the probability. So I hope this is not too complicated for people, but essentially, like the second one is only valid when you fix n in advance, and the first one is valid, simultaneously for all n from 1 to infinity. So and so this, if you build the usual pointwise intervals, they're not valid time uniformly. So if you take this L tilde, U tilde, which has this type of a guarantee, and you ask, is it uniformly valid? And the answer is not. It's like you want a 95% guarantee. Let's say alpha is 0.05. You want a 95% guarantee, you're actually going to get a 0% guarantee. So this is typically the case. And so you really don't want to use intervals that were designed for fixed times. You do not want to use them sequentially. You will make an error. And with probability 1, you will make an error. And so, so the confidence sequence is the right notion to be using sequentially. So this is like tracking the mean of a Gaussian or a Bernoulli from IID observations. If I draw x1, x2 from a normal with mean theta and variance 1, and I ask you to track the mean of the Gaussian over time, how do you do that? Or if I give you a coin and I tell you, keep tossing the coin, you get heads, tails, tails, heads, heads, tails, and you keep seeing the data that you're getting. And I ask you to come up with an estimate of the mean and a confidence interval for the mean as you collect more and more data. How do you do that? So basically producing a confidence interval for these, in these settings is elementary. But my question I'm asking is, how do you produce a confidence sequence, which is like a confidence band over time? So visually, it's going to look something like this. So let me just take you through this. On the x-axis is the number of samples in log scale. Okay, so you could think about this as a number of coin toss. So what I'm doing here is I'm tossing a fair coin. -1 is tails, +1 is heads, 0 is the true mean. And so I'm tossing it 10 times or 100 times, 1,000 times, and so on. So it's just log scale number of samples. And here are the confidence bounds. The empirical mean is in the black line. So obviously, even though the true mean is 0, when you actually toss a sequence of coins, the empirical mean is not going to be exactly 0. It's going to be a little bit away from 0. Fine. So that's what you get. That's the empirical mean in this run of the experiment. If you use a pointwise confidence interval, for example, something like the central limit theorem gives you these, or if you have binomial confidence intervals, that's what's in the green dotted line. That's what's being plotted. And these are not sequentially valid. These are only valid at large n. If you fix it in advance, then it's valid. It's not valid sequentially. And then there's this anytime confidence intervals or confidence sequences, which are in pink, and these are valid sequentially. And so if you want one example in this run, and it will happen in every run, but one example in this run is if you look at time 140 or so around over here, the true mean is 0, but the confidence interval given is something like 0.02 to 0.3. So it does not contain 0. So given by the central limit theorem, it's just wrong here. But the pink one is actually still correct here. These are not actually this is from an older paper of mine. These are not the best bounds one can get, but they're a little bit wider than the central limit theorem. But they're the guarantee of the green dotted line is point-wise valid at a fixed time. The guarantee of the pink line is that this gray line will be inside this pink band uniformly from 1 to infinity. It will always be inside this. In this case, the gray line is trivial, it's at 0, but imagine that you were looking for small effects, and so maybe the gray line is actually at 0.02 or something of that kind. And so this pink line will always contain the gray line, the true mean, from time 1 to infinity. And so what you see basically is that small sample sizes, like this is about sample 30 or something of that kind, the pink line is very broad then, and perhaps it should be broad then. You really don't have much data to make any inference. But already at time 100, it's just a little bit wider than the central limit theorem. And by time 1000, it's very, very, very slightly wider than the central limit theorem. So essentially, if you're collecting enough data, like more than 100 samples, then there's not much of a price to pay in going from a fixed time confidence interval to a confidence sequence. But the confidence sequence gives you a much stronger statistical guarantee, which is that you can continuously monitor your data as you come in, and you can stop adaptively and no problem. So that's the advantage of a confidence sequence or an anytime confidence interval. This is just one example of what it looks like. Again, this is not the best one, but I just want to give people some sense of what is that thing that I plotted. So it's centered at the sample mean. So this pink band will always be centered at the sample mean, and then plus or minus some quantity. Okay, so that quantity, it shrinks with n at a square root 1 over n kind of rate. If alpha is 0.05, then you get some log 1 over alpha kind of dependence, and then there's this log n kind of depth, and the constants are very small. So this is an example. This might seem mysterious, but I don't have the time to describe where it comes from. This is a theorem that if xi is actually Gaussian, let's say, or it's bounded between -1 and 1, then this will always contain the true mean mu from 1 to infinity. So mu will lie inside this for every n. You can plug in any n and it's fine, simultaneously for all n. So some implications of having a confidence sequence. So this is a slightly different definition of a confidence sequence. This is, if I take the definition I gave you, which is a positive statement, I want theta to be in the confidence interval for all n. Now I'm negating that statement and writing it just so that you get used to seeing different statements. Now I'm saying, is there some n for which theta is not in ln/un? So this is an error statement. I'm unhappy if this happens, and the probability of this is at most alpha. So I've just taken the earlier statement and negated it to write it differently. This is what a confidence sequence guarantees you. And so some implications of that is actually that it provides valid inference at any time, even stopping time. So for any stopping time tau, the probability that theta is not in L tau U tau is at most alpha. So now tau is not a time you fixed in advance, like 100 or some 1,000 or whatever. It's a data dependent time that you decide on as you see the data. So essentially, in this case, the interval is based on x1 to x tau. That's the data you've seen so far. That's what the interval is based on. And tau depends on x1 to x tau. You chose to stop at time tau because the data x1 to x tau seemed promising. So that's the part where it's different from a confidence interval. In a confidence interval, the n is not allowed to depend on the data x1 to xn. x1 to xn has to be just independent of n. N has to be chosen in advance. But here, tau can depend on x1 to x tau. You can monitor, you can stop, and even then, the interval that you have at the stopping time is actually going to be correct. So that's a very powerful statement. It also allows post hoc inference. If your boss comes and asks you, hey, you ran this for 10,200 samples, why did you not stop at 8,000 samples? Or why didn't you stop at half the sample size? Then actually it turns out you can answer post hoc questions, which is you can say in hindsight, for any random time t, which is not a stopping time, also time picked in hindsight, the probability that theta is not in LTUT is at most alpha. So this is again a random variable. Tau and t are random variables. And in general, there's no pre-specified sample size. You can extend or stop experiments adaptively. So this issue which I had mentioned earlier, that issue arises because people are taught and mostly use confidence intervals. But if you switch to using confidence sequences, then it takes away that problem. Okay, so these are papers titled, if you want to look at it, it's time uniform, non-parametric, non-asymptotic confidence sequences, and meaning that all of these bounds are non-asymptotic. We don't rely on n being large for our bounds to be correct. They are derived under non-parametric conditions. I don't need your data to be Gaussian or Bernoulli. I just demonstrated it for that purpose, but they can be derived under non-parametric conditions, and they're time uniform. They're valid over time. And this should be contrasted with what is usually found in a textbook, which is it's only valid point-wise, not time uniform. It's usually parametric, and it's usually asymptotic, relying on something like the central limit theorem to derive a confidence interval. So we suggest moving to things of these kind, which is technically safer in practice. Okay, so I will probably stop this for the reasons of time. There's many extensions in the paper to continuous time processes, to matrices, sequential average treatment effect estimation, which is in a causal inference language, sequential covariance matrix estimation. There's many extensions of this, but I presented the simple. I'm tracking a single scalar over time. But if you want to think about other extensions, please have a look at the paper. Okay, so that ends.
Sorry, Aditya, can I ask a question briefly?
Yeah, absolutely. Yeah.
And in the previous slide, the second example, continuous time process, what would be the specific application here? Is it for
this is for finance. So typically in finance, the data that they come in, stock prices, things of that kind, they typically model that using continuous time processes, not discrete time. So that's the setting. Just because of the frequency of the data coming in is so high, and the changes in the prices are, well, the earlier theory was that it looked like a Brownian motion, and so they would use continuous time processes to model financial stock price changes. So this is a different sequential setup. If I was giving a talk in a finance company, I would maybe highlight that more. But I don't know, in tech, I mean, I can't imagine. Maybe there is such high frequency data, like in thousands of changes per second, kind of, if you're collecting data of that frequency, then thinking about things in continuous time might help. And so we have such boundaries for sequential estimation in continuous time as well. So great question. Thanks.
Yep, thank you.
Maybe a slightly different one is there's a Poisson process. So there are things where you don't know if the event is happening rarely, and the event doesn't happen at regular intervals. There's just some event that happens at some time, and then at some other time. And so you can think of that as a Poisson process if you're tracking these kinds of rare events that happen once in a while, and you want to study it. Then it might be better understood using a continuous time Poisson process.
But then for the Poisson process, the confidence intervals would be there for intensity of the
exactly.
process. Okay, exactly. That's kind of what I was trying to grasp.
Right.
Thanks.
Okay, so I'll now move to, that was for the inner sequential process, which is one experiment. Now let me zoom out and go to the outer sequential process. Let's say we are now doing things correctly within each experiment. We're using a confidence sequence, so hence we have a correct confidence interval and things like that whenever we stop. So the inner sequential process is correct. What do we do in the outer sequential process? Okay, so again, this is based on a series of works, which is again a mix of machine learning conference papers and statistics journal papers, with a bunch of excellent students, collaborators, postdocs of mine over time. So I'll probably share these slides with Jeff so that you can go through it. But again, if you go to my website, you'll be able to find a section just on online multiple testing, which is what this area goes under. So the first part I'm going to mention is, there's something called the false discovery rate, which is an error metric in multiple testing, and how do you control that in a sequential fashion when you have independent p-values? And then I'll just mention that there are extensions to other error metrics, false coverage rate for intervals, or how do you handle dependence and so on. So I'm going to start with the simplest setting. You're doing a sequence of tests, different tests. You're testing different hypotheses over time. You have independent p-values, and you would like to control this error metric, which is called the false discovery rate. Now, what is a discovery? It's like a discovery is saying, oh my God, I found something in this experiment. This change will improve our money or our looks or our time spent on the website. It will improve something. In the sciences, it would be saying that this gene is related to diabetes, or this part of the brain processes language, or something like that. So the notion of a discovery depends on which field you're in, but it's basically saying, oh, I found something interesting that people didn't know. That's a proclaimed discovery. And the numerator is a false discovery, which is saying there's nothing going on. What you found was pure coincidence. There was some chance fluctuation. You didn't correct for it, and so you think you found a discovery, but there was no discovery to be found. So that's a false discovery. And so what you want is maybe you test thousands of things. Okay, let's say you have 3,000 tests that you run over a year. Maybe you proclaim 300 discoveries. Maybe you say over time, these 300 changes would have helped things. You would like the false discoveries to be maybe 30 or something like that. Maybe you would like it if 270 of the changes that you make are good changes and only 30 of the changes that you make are bad changes, or they had no effect at all. You would like that to be the case. Okay, so the question is, how do you ensure that? How do you control the false discovery rate? Okay, so again, I said the reality is that typical internet companies run many different thousands of tests over time. You might say, oh, does changing the color, does that affect something? And your decision rule might be, is the p-value for that test, is it less than alpha? Then, okay, you say, oh, well, maybe if I change some icon, then more people click on it. Well, you'll test, is the p-value less than alpha? And so on. So, point is that this will continue forever, and people will keep coming up with ideas of things that will improve whatever metric they're interested in. And if you keep ignoring the other hypotheses and you're saying, like, oh, like I'm going to test if the p-value is less than alpha and I'll make my decision based on that, that's a problem. Okay, so what is the goal? The goal is, given a possibly infinite sequence of tests, basically given these p-values for each test, and these p-values are now correct and they're valid under optional stopping and so on, you can move between confidence sequences and anytime valid p-values. We show how to do that in the paper. So confidence sequences can be used for deriving anytime valid p-values. So you've done this correctly, but now you have many tests. How do we control the FDR in a fully online fashion? And the answer is going to be, I have to change the level at which I'm testing. So I have to test each PI at a different level. And the question is, how do I set this error level so that the goal is that for every time t, the FDR up to time t is less than alpha. So the number of false discoveries I've made in my first t tests is at most alpha, but this is for any t, because I'm by June I've run 300, by August I've run 600, by December I've run 800. Like I want this guarantee forever. I want this forever. I want to know that I'm keeping the number of false discoveries somehow in check, under control. Now the important part is that in the decisions are irrevocable, meaning that you make a decision when you run your test, you look at the p-value, you make your decision. Maybe you make your change to the website, whatever, and you move on. It's not like 3 years later you go back and you reverse that. I mean, maybe you will reverse that decision, but we will still, if it was a bad decision, we'll still count it as a false discovery, because it might hurt to take an action on that decision. So we make a decision as soon as the test. We don't make a decision 6 months later or 1 year later. We immediately decide this is what we want to do now, not knowing the future. That's the important part. We have no idea what the future is going to hold, what p-values, how many tests, no idea. And the idea is that typical offline FDR methods, if you've heard of the Benjamin-Hochberg method, things like that, they do not have this guarantee. So if you run the offline FDR method on the first 5 hypotheses, and then the first 6, and then the first 7, and then the first 8, if you keep running it in that fashion, it will not have this guarantee, because it will keep changing its decision. Meaning, when you run it on the first 3, it will say reject the second one. Then you run it on the first 4, it might say don't reject the second one. When you run it on the first 5, maybe it will say reject the second one. And so you're like, the offline methods are not intended for this purpose. They're just intended for a scientific setting where you have 10,000 hypotheses in a batch. You correct them all in one go. But what we're going to derive are online false discovery rate methods that don't need to know the future, can make irrevocable decisions as soon as the test happens, and yet have this guarantee over all time. That's what we're going to be able to achieve. And the idea is very simple. So I'm going to first tell it to you pictorially. You have an alpha or an error budget. This is the alpha that you start out with. It is saying that I want my FDR to be less than alpha. That alpha could be 0.1, let's say. Or if you're willing to tolerate 1 in every 10 decisions that you make is false, okay, maybe that's okay. Then alpha is 0.1. So you start off with a budget of 0.1. And every test uses some of your budget up. Okay, so every time you start a test, you cannot run it at alpha. You cannot run every test at that same error level. So you have to use up some of your budget whenever you run a new experiment. But now this seems hopeless because if I run too many experiments, I've lost my budget. So it turns out that every time you make a discovery, you actually earn back an extra alpha. So the budget or the money that you have for running experiments now, this is like the alpha budget, meaning the error budget, that can actually increase beyond alpha. And then you spend it over time. So basically the error budget that we will have for each experiment is data dependent. It depends on what has happened so far. As long as every once in a while you make a discovery, you will get back error budget that you can then spend in the future. So that's what the process is going to look like. It's not going to be a decreasing sequence of alphas, and it's not going to be a fixed alpha. It's going to be, it's going to decrease initially, but increase every time you make a discovery, and then decrease and then increase every time you make a discovery, and so on. So that's how the algorithms are going to work. Okay, so this was conceptualized, 13 years ago in an idea called alpha investing. That made some progress over the last 10 years. There was progress on this. I would say it culminated with the Annals of Statistics paper with something called the Lord algorithm. And they were the first ones to realize that, oh, this can be useful for A/B testing. Like the previous applications had a little bit more of the sciences in advance and something called a quality preserving database that you can make multiple queries to over time and so on. But, these authors realized, Javanman and Montanari realized, oh, it can be used for A/B testing kind of settings as well. But then actually the Lord algorithm was the state of the art, but then we immediately found that we can improve strictly on LORD. So we wrote LORD++, which was a strict improvement of the LORD algorithm. And then this was in NeurIPS and then ICML, and then the most recent NeurIPS, a sequence of algorithms that progressively improved on LORD++. So Addis, it's called adaptive discarding. The Addis algorithm is, I think, the current state of the art, but I will not go through the details of each algorithm separately. So there are two viewpoints. The earlier viewpoint, was an algorithmic viewpoint. It said we have to specify this algorithm. I showed you this cartoon figure of your wealth that you spend and you receive, you get wealth back on a discovery, things like that. I have to specify how much wealth we start off with. I have to specify how much wealth is subtracted every time we make a test. And I must specify how much wealth is awarded for each discovery. So that's like a very algorithmic viewpoint for how to design an online FDR algorithms. And then, you prove that for these choices, the FDR is actually controlled. Okay, so that was the earlier literature. But then we switched it in order to discover new algorithms and better algorithms. We switched it to a statistical viewpoint. We said to define an algorithm, what we're instead going to do is we're going to define first an estimator or an empirical estimator of the false discovery proportion. So actually remember that we don't actually know which discovery is a true discovery or a false discovery. We have run a test, we make some decision, but we don't know whether that decision is a correct decision or not. The data that we've collected seems to tell us that, oh, maybe there's some effect here, but it could be a false discovery because we're testing thousands of things and you might observe things in the data just by chance. So it could be a false discovery. So we do not know the false discovery proportion. We do not know which of our discoveries are true and false discoveries. So, but we can use the data that we've collected to design an empirical estimator of the false discovery proportion. So that's FDP hat. We don't know the FDP, but we know FDP hat. And we prove a meta theorem that says that if we keep the estimated FDP less than alpha, then the true FDR, which we don't know, is also less than alpha. So we prove this kind of a meta theorem. And then we design certain what are called monotone sequences alpha T, alpha 1, alpha 2, alpha 3. We design certain monotone sequences alpha T. They're not monotone in the sense that they're decreasing or increasing. They're actually monotone in the discoveries that you make, which means that every time you make a discovery, alpha increases. So that's it's monotone in that sense. And so we design monotone sequences that ensure that FDP hat is always kept less than alpha. And so the new algorithms were based on a slightly different statistical viewpoint. Okay, so it helps us design better new algorithms by focusing on estimators, being like, how do I design a better estimator of the false discovery proportion? Okay, so this is what an oracle estimate of the false discovery proportion looks like. You're not an oracle, but if you were an oracle, this is something that you would do. The denominator is just the number of discoveries. You know this. You know the number of discoveries because you know which things you acted on and reported and proclaimed and so on. The numerator is the number of false discoveries. That's what you don't know. You don't know which of the discoveries are true and false. So instead of that, you plug in the sum of the alphas that you used to test the nulls. Now, you don't know which the nulls are, so you don't know the numerator simply because you don't know which the nulls are. But if you keep this FDP star, this oracle quantity less than alpha, then a certain quantity called the modified false discovery rate is also kept less than alpha. And if the p-values are independent, then the FDR is also less than alpha. So this is kind of a meta theorem that says if you were an oracle, this is how you would estimate the false discovery proportion. But you're not an oracle. So how do you do this? So one way is you just drop this. The sum says, I'm only going to sum over the nulls. The nulls are things where there's no discovery to be made. I don't know which those are. So instead, I'll sum over everything. I know the sequence. I set the sequence of alphas. So I know the numerator and the denominator. So that's my estimate of FDP hat. So the algorithm lot++ just says, keep this FDP hat less than alpha. Set your sequence of alphas in such a way that this is always kept less than alpha. So every time the denominator increases by 1, because you make a new discovery, now the numerator can increase by alpha. So you have an extra budget to spend in the numerator. That's why the algorithm works out the way it does. So then, for saffron, I won't go into this, but we define a better estimate of their false discovery proportion. There's some default values you can try out. And then we improved it even further by saying, well, if the p-value is large, just throw it away. Otherwise, if it's small, then double it and test it. So it's like something called a discarding trick. And so so there's a sequence of steps to improve the false discovery proportion estimator because we don't know the truth, but we can estimate it. So you can extend this, that's the estimate. It's very simple. The alphas, the discoveries estimated, keep it less than alpha. You're done. That's the algorithm. And just do that instead of setting everything at level alpha. Just do it, set it adaptively, and you have FDR control. Now the extensions that we have are people don't appreciate this, but they think multiple testing only applies to testing. And if we moved away from testing to confidence intervals, somehow the problem would go away. Now, the problem doesn't go away. If you have many confidence intervals that you're tracking over time, like different tests for different parameters, you're tracking different confidence intervals, but only acting on some of them. For example, only if the confidence interval doesn't contain 0, then you will proclaim this drug to actually work, or you proclaim this change to actually work. So it could be not test-based explicitly. Even though there's a duality between tests and confidence intervals, you might think of things in terms of confidence intervals. The selective reporting or acting on confidence intervals also inflates not the false discovery rate, but the false coverage rate. And so the idea is that, like, if you're testing the first drug and you have some treatment effect and you get a confidence interval like -0.1 to 0.2, and you test the second drug and you get some other confidence interval, you test the third drug. So there's many drugs that you're testing over time. And then finally, you find a drug which is where you find that the effect is between 0.1 and 0.2. Like, that's the confidence interval does not contain 0. You think it has a positive effect. So you choose to act on that drug. You say, okay, great, let's run a much larger clinical trial on it, or just let's roll it out, or whatever it might be that you do when you make a discovery. And this it doesn't have to be a drug, as I've said, could be your own A/B test. This is a problem because every test is being run at a 95% level. And you can prove that even if everything was just null versus null, meaning control, and control. Suppose I only had gray pills in this whole thing, everything's a control. Even then, you will discover things. Every once in a while, you'll say, hey, I found a drug that works. And so that's terrible. That the reason that's happening is because you're using the same alpha and you're not correcting for multiple testing. And so the false coverage rate is actually the expected ratio of the number of incorrectly reported confidence intervals, which you don't know, to the number of reported confidence intervals that you do know. These are like your denominators, again, like your discoveries. You go and tell your boss, hey, I found some effect and here's the confidence interval for that effect. So if you only report certain confidence intervals, some fraction of them are going to be false, and you want the false coverage rate to be low. And the same the same question we asked for testing, you can ask the same question for estimation. And so when experiment J starts, we must assign a target confidence level alpha J, which is not alpha for every experiment. And when experiment J ends, we must decide, do we want to report theta J and the confidence interval for it? And this must be done at any point in time so that the false coverage rate is controlled. And so we had an ICML paper last year, which basically extended the FDR algorithms to this kind of a setup. And it's important to remember that FDR control does not imply FCR control, but the implication moves the other way around. These are my last couple of slides. You can also, you might say, I don't want to control things only in expectation, like the false discovery rate and false coverage rate are expectation bound, meaning it says the expected value of this ratio of false discoveries to discoveries is less than alpha. You might say, I want a high probability bound. And so we can give high probability post hoc bounds on the false discovery proportions. You have run a sequence of experiments and you can look back in hindsight and say, this is a valid upper bound with probability at least 1 minus delta, the false discovery proportion, the random variable, the ratio between the number of false discoveries to total number of discoveries. This is always less than this quantity. This is the number of rejections you've made. These are the alpha levels you gave for each experiment, and this is the level of confidence you want. So this is a simultaneous bound over all time, and so it can be looked at post hoc if you want to look at what happened. This is valid, not just in expectation, basically. And this is true for the false coverage proportion as well. The last thing I'll mention is that you don't have to keep track of all of time. You might say, look, I'm in 2024. I don't care what happened in 2021. It was so long ago that those false discoveries should not affect me today. And so you might want to smoothly forget the past. Recent tests might be much more relevant than old tests when you're looking at how well you're doing in terms of false discovery rates. You might want to forget the past. So, we define something called the decaying memory false discovery rate. So you choose a decay like D less than like 0.99. If you chose 0.99, and what you did was you weighted the discoveries exponentially in time so that older discoveries count for almost nothing and the newer discoveries are weighted the most. So then this basically by about 1 over 1 minus D steps, it kind of forgets what happened before that. So we have algorithms that can control the decaying memory FDR if that's a more sensible error metric. It's like a moving window FDR or something of that kind, but it does this kind of smoothly. So we have algorithms that can control this instead if this is a more sensible error metric for if a company deems this to be more sensible. So there's software for a lot of this. There's an R package currently called Online FDR that's been maintained by David Robertson, who's a postdoc at Cambridge in collaboration with me and several others. There's going to be frequent updates and wrappers in the months, but a lot of this is already implemented online. So if you use R, you can already download and try it out. If you use Python, there might be wrappers that go in between them, or we can have help in updating all of these and importing into Python as well. I think we might have it in Python as well. You can find links to all of the papers that I mentioned on my website. So let me stop there, acknowledge funding from my career grant at the National Science Foundation. And thank you to all of you for staying, and I'm happy to take any questions.
All right, thank you. That was excellent. I guess we can start with a question. So in terms of the online sequential experimentation and just multiple experiments on the same data set and the analogies that you gave at the start, often in science, you would see a data set published, like ImageNet data set, and then everyone would use that data set, which is, that's fine because that's like a benign computer vision algorithms being trained on it, although there are some ethical considerations with it. But you also have challenges in medical domain where someone will publish a protein data set, or someone will publish some sort of brain scan data sets, and then people try to achieve some sort of discovery or who will perform the best. Are you familiar with these kind of methods being incorporated into these competitions or into the evaluation over these data sets?
So actually not. So I should, that's a great question. I don't know how to handle that situation yet. So in general, I think that's an open question for the field, which is, with these types of, one of the questions I posed at the start is, does the data set get stale, and how do you know it's gotten stale, and so on. I think that's still pretty much an open question. The setting over here was actually this independent p-values for the different hypotheses. And so the setting I'm imagining is really, you have a different drug in each round, and you have different people in the clinical trial. And so the focus is just on the multiple testing aspect. So even if there is no dependence between the hypotheses, and no dependence between the data, and no dependence between the p-values, even if there's independent tests over time, there is still a multiple testing concern, which is, if you just test enough things, some of them will appear to be true just by chance. And so you'll reject hypotheses just by chance and make discoveries just by chance. And how do you correct for that? And so this multiple testing talk was just focused on that aspect of it, and the aspect in which the data is actually fixed and the hypotheses are changing over time, and you're making more and more queries over time. This is actually very subtle, and I'm actually working on differential privacy techniques to make adaptive data queries to a database in order to be able to handle this type of a situation. So you can't, if I just have unfettered, unrestricted access to a database and I can ask any question I want and any number of them, there's no hope. So there has to be some way of saying, I'm not going to release like too much information, which with every hypothesis that you're testing, I won't release too much information. So another way of saying it is, I release a noisy p-value from that data set. And I don't give you access to the data set. You tell me what hypothesis you want to test. I will tell you back a p-value, but a noisy p-value for it, so that you can ask many more questions from that same data set and so on. So there has to be something like that that's done, so that the validity is maintained.
Mm-hmm. Sounds very interesting. That sounds like something that organization like Kaggle would definitely want to adopt in terms of their competitions.
Yeah, absolutely. Yeah.
All right. And the other question is coming back to the topic of the presentation itself in terms of multiple hypothesis testing, be it A/B tests or drug design. In the last decade, or maybe before, the tech companies have been very much pushing the idea of experimentation culture. They build the platform that is very much usable to do A/B tests. Anyone can run the A/B tests, and they encourage everyone to run A/B tests. And it's almost as a metric, how many A/B tests did we run this year? And they want to run as many as possible because they associate that with the improvement. From what I've seen, I haven't seen these methods used in practice in terms of false discovery rate control. And are you familiar with any studies that try to see if what are their false discovery rates in these organizations? Or that's one side of the question. And the second one, are you familiar with these methods being used in practice in these organizations where they have experimentation culture, they run a lot of A/B tests, and then they try to have some implicit algorithm or explicit algorithm underneath that tries to, okay, you want to run the algorithm today, well, you get this much of alpha. You're running the algorithm A/B test there, you get this much of alpha.
Again, a great question. I'm not aware of companies doing this. Part of the reason is some of this is extremely new work, meaning the last 1 or 2 years, and I don't know that companies are aware of the published work yet. And another one is, like, I think online multiple testing is just a problem that, yeah, I would say, like, it's not that they're not aware of the solution. I think most people are not aware of the problem, because when hypotheses all appear in a batch, people are trained, okay, I have to correct for multiple testing. But when they appear one at a time, people are just they don't think in the same way, and they don't realize, oh, it is an issue I need to correct for. So in some sense, there's some convincing that needs to happen that the problem itself exists before here's the solution to that problem. And so I think that will just take time. I do, have given talks about this at various companies, Microsoft, Amazon, like now Winter, things like that. But I don't know if, because a lot of it is proprietary, I don't know what they're actually doing at their backend now. They, after my talk, has anything changed or not? I have no clue. And I think it's just proprietary, and they don't release the data, so I can't check post hoc, how things are going. I don't have access to it. Maybe if I actually moved to a company, then I would have access to such kind of data. But currently, I don't. So I'm sticking to giving talks at companies and hoping that at least some of the ideas, if not literally the same algorithm, some of these ideas permeate and people start to adopt better statistical practices.
I would imagine certainly drug design companies would be very interested in these sort of things, because there it's a very high stakes decisions being made on the
Yeah. So
test.
actually, I have given a talk at a pharmaceutical company called Berry Consultants just a few months ago, and yeah, they were interested in it. That's why they invited me to give the talk. But I don't know if they have incorporated it or not, because in some sense they have these long established practices, and it just takes time to change an organization. It's not a flip of a switch, something of that kind. So because a lot of it is FDA monitored and things of that kind. So maybe do we have to convince the FDA before the companies are allowed to make this type of a change to their setup? I'm not sure. So great question though.
Thank you. And another one in regards to the multiple hypothesis testing or sequential testing. A lot of the A/B tests that are being run, you can almost do a clustering of them, right? So one particular team will do A/B test that is related to this particular hypothesis, and they will keep developing around that particular idea. And then totally unrelated team will be doing some other hypothesis testing. And in some of the algorithms like LORD, for example, and other algorithms you've mentioned, you have to define the wealth for the testing. And I wonder if that wealth and has to be defined per that family of ideas or hypotheses that you are testing, or does it have to be global? Kind of abstract question.
Yeah, this is again an excellent question, which is similar to the question of what scale should we be correcting for multiple testing at, like within a single paper or a lab or this. And so like I think for each organization has to make that decision to should there be just to pick something other than vintage, should there be a Google level control, or should there be a marketing team control and a Chrome team control and a cloud team control and something else? Like that's a decision that an organization has to make. And then even maybe there could be sub teams within teams that are doing different things even within Chrome. And so like I don't know the answer, because this is like it's just a choice that that company has to make. How much basically I think the tensions are if it's too narrow granularity, then there's a huge multiple testing correction that you're not accounting for. There's just like hundreds of teams and everyone is kind of separately. It's like the thing where you have 100 hypotheses and you just test one every day and don't correct for the 100. It's like if there's 100 teams and you're really not correcting for that fact, and each team is correcting within itself but really not across, then you're in trouble. But on the other hand, if you're trying to coordinate things across 100 teams, the teams are going to get upset because they're going to say, wait a minute, why is my alpha different because some other team ran some tests that I don't approve of, and now my alpha is different? And so how do you balance the incentives at the team level from the incentives at Google level? Google does not want random tests to come up with spurious claim discoveries that are false and then to invest money in changing its system so that test. So like Google cares that, oh man, in its organization. So for example, Microsoft runs about 36,500 experiments a year, like about 100 experiments like a day. That's the number of experiments that they're running at the Microsoft scale. It's a similar number at Amazon is running thousands of experiments at any given time. They're running thousands of A/B tests in parallel. And so the question is, like, these organizations are going to be and should be concerned that lots of these are going to lead to spurious discoveries and then changes made to the system because they think they're doing something better. They should be concerned about that. But like the team doesn't care about what the other teams are doing. Each team might think they're making really good discoveries and testing really good hypotheses, and the other teams are wasting their time. Their team is doing something sensible. So how do you balance the team level incentives from the company level incentives? That's a difficult question. That's an incentive, like almost a sociological question, and not a, maybe it's partly a mathematical question. Maybe there's a framework that can balance both incentives at the same time, but currently I don't have that. It's a great question to think about next step.
Yeah, that sounds fascinating. It kind of reminded me of a Chinese restaurant process where you kind of try to cluster adaptively things. And I wonder if you could build a simulation to actually test these ideas somehow. Not sure, but maybe that's something could be explored later.
Yeah, yeah, great question again.
We have a comment, actually. Let me read it, and we'll see if it's a question or if it's a statement. So when FDR is being controlled online, is it true that a particular A/B test is more likely to be a success if it is performed after a discovery?
Yes. So I guess this is related to my first question which was in science, does the importance and relevance of discoveries depend on the order in which they are tested? And I believe that is the case in science, and I believe that is the case in tech, and that is the case in these online FDR algorithms as well, is that the order of the algorithms definitely does matter, but that still maintains the false discovery rate. So typically we say if you have some prior knowledge of which hypotheses might be more likely to be non-null, you're not sure, but more likely to be non-null, you might want to test those first. And then things that might be high risk, high reward, meaning they're not really likely to be non-null, but if it was, it would be great. Then you want to run it after a discovery and so on. So like the order matters, and thinking about the order will actually help if you have some prior knowledge. So that's a great comment or question, and the answer is yes, order matters, and yes, you should exploit it to your benefit, and so on. Yeah.
All right, and there is a follow-up comment. Thank you. If so, is there a risk that different teams that are motivated to run successful A/B tests will wait for discoveries to happen before running their own experiments? Kind of tying out back to the previous question.
Yeah, that's a great question again. Yeah, I don't know. So like this is again a question of incentives. And I haven't addressed incentives at all in my talk. And I don't know what's the right way to address that incentives question. You're right, that this creates an incentive to be like, let's wait for the, like, I wouldn't say it's an obvious incentive, because if you wait for the other teams and they're testing nulls, they might just lose. They might just keep losing well, and then maybe you're just left waiting, or and then you finally say, okay, I'm going to stop waiting. I've wasted a week already. Let me just run my test now. Now you're running it at a much lower. You can't see the future, so you don't know when the next discovery is going to be. So if the next discovery is going to be a day from now, maybe you can wait. If the next discovery is going to be 3 weeks from now, maybe you don't want to wait. And so you don't know the future. So I'm not sure you can game it that much. But I admit that there is this incentive question. There is this incentive to let's wait a little bit to see if the next one and run it next after that. And I don't know what's the right way to account for that balancing of those incentives. I mean, the FDR at a company level will still be controlled, but maybe some other metrics might not be controlled. Or maybe, I don't know, like satisfaction of these teams, or something else might not be controlled, but the FDR will still be controlled no matter what the order is, because the order is still data independent in the sense that the data that you're getting to test this hypothesis is fresh data. No matter when you tested it, it's still fresh data. It's an independent p-value, and that's all that's needed for the FDR control guarantee. But I can see the issues that arise, and I'm not sure what's the best framework to. I can't even crystallize what is the problem. I can see that there is this incentive issue, but so what? So maybe we have to think a little bit more of what the problem is and be able to write down the problem, and then maybe we can try to formulate a solution based on some kind of incentive compatible schemes or something of that kind.
Yeah, excellent. Sounds like we need to formulate a whole new problem.
All right. Yeah, there's plenty of research to be done on this. I think this is like the start, but I think doubly sequential experimentation is a right field. I mean, I have a few papers on it, but this is what's going on. People have been focusing on either the inner process or the outer process, but really we are running a sequence of sequential experiments, and I think progress on understanding that, like what that creates, like this viewing in that way, what problems does it create and what solutions does it require, is an interesting ripe area for future work.
Mm-hmm. Amazing, excellent. So I think given the time, we should be going towards wrapping it up.
Sure.
So again, thank you so much, Aaditya, for being with us today. We'll be posting later on some of the links to your website, to the papers that you've mentioned today, and naturally, if you share your slides, we'll be sharing that as well. So thank you to you, and thank you to everyone who joined us today.
Thank you, my pleasure.
All right. Okay, I believe we've finished.